Hi guys, welcome back to my channel BioLounge. Let's get started. Now we are going to learn about antibiotic resistance as selectable marker. So first of all, you should understand why we are going for a selectable marker. Okay, so let us analyze this. Okay, one is traditional method of hybridization. What does it say? Traditional method of hybridization says that when you are crossing two plants, what happens is that both desirable and undesirable genes are getting inherited. So that is not the purpose of doing this process. Why? Because it's going to be a very long process and lot of energy is needed for that, and undesirable genes are also taken into account in this type of hybridization. Whereas in biotechnology and genetic engineering procedures, we are actually propagating only the desirable genes and we are going to eliminate the undesirable genes. We are going to select only the desirable genes and the undesirable genes are eliminated. So how are we going to do this process? We are going to do this by means of a selectable marker, that is antibiotic resistance. Students first understand that bacteria such as E. coli does not carry any antibiotic resistance gene in it. Please understand that first. There is no antibiotic resistance gene in E. coli. So what we are going to employ here? We are going to employ an artificial plasmid. Plasmid, you all know, they are extra chromosomal circular DNA. They are capable of independent replication. They are not dependent on the host DNA. So this plasmid has been constructed artificially. So P stands for plasmid, B stands for Bolivar, R stands for Rodriguez. So these are the two scientists who were involved in creating or constructing this first artificial plasmid. What was so special about this is that this plasmid contains two regions. This region contains a gene that is responsible for resistance to ampicillin. So you write AMP capital R. This region that you see here carries resistance to tetracycline antibiotic. So it is TET-R. Now what do you mean by this antibiotic resistance? See, when you are sick, okay, for example, you are affected by a bacterial infection. So the doctor will prescribe an antibiotic that is exactly going to kill the bacteria that has entered your body. So antibiotics are made, they are prepared just to kill the bacteria. Now you are creating a plasmid here that contains a gene that is going to provide resistance to ampicillin and tetracycline. Suppose you are going to introduce this into E. coli. E. coli does not carry any antibiotic resistance gene. That means you can kill this E. coli very easily by sending an antibiotic. Now what you are going to do in this method is, you are going to send an ampicillin resistance and a tetracycline resistance gene into E. coli. Thereby the E. coli carries what? Antibiotic resistance in them. So this is the purpose of doing this experiment. Now let me tell you once again, traditional method of hybridization failed because undesirable genes also were propagated along with desirable genes. That is point number one. Point number two, we have an advantage in biotechnology. We are selecting only desirable genes and only that will be propagated in large numbers. Undesirable genes are eliminated. Point number three, for that we are constructing an artificial plasmid called PBR322. Point number 4, in this plasmid you have two antibiotic resistance gene. One is TET-R and one is AMP-R. Now, E. coli does not carry any antibiotic resistance. And point number 5 tells you very clearly that E. coli does not carry any antibiotic resistance gene in them. Now this forms the introduction. Let us go into the concept now.